It's time for the 3-question warm-up for Foundations 4. Let's get going. First question here, which amino acids are found in the nuclear localization signal? So this is the lysine, arginine, and proline. Next question, what is the most common cause of intellectual disability in infants? The answer is fetal alcohol syndrome. Next question, how do RB protein and P53 regulate the cell cycle? Well, they prevent the cell from progressing from G1 phase to S phase. Okay, that's it for the 3-question warm-up. Let's get to that lecture.
We're still talking about cell biology as a foundation for everything that's going on in the body. We've talked about the major organelles, and in this video we're going to focus on the cytoplasm, which contains structural components and a cytoskeleton, and also enzymes and storage molecules. So first let's talk about the different structural components of the cell. Cells aren't just bags of intracellular fluid, right? They have specific shapes and a specific orientation in space depending on the specialized function of the cell. And by and large, it's the cytoskeleton that gives the cell that shape. There are 3 basic types of filaments that make up the cytoskeleton. There are microfilaments, which are the thinnest filaments. There are microtubules, which despite also being called micro, are actually the thickest of the 3 filaments. And then in between the microtubules and the microfilaments are the intermediate filaments, which have a diameter about half as large as the microtubules. And there's also myosin, which is a little bit larger than the intermediate filaments. So again, going from the thinnest to the thickest, it's microfilaments, intermediate filaments, myosin, and then microtubules. So microfilaments are made of polymers of a protein called actin. Now actin is really important for cellular motility, whether you're talking about neutrophil chemotaxis or macrophages or the microvilli of the intestinal brush border or activated platelets changing shape to plug up a bleed. All those kinds of cellular movements are mediated by the growth and contraction of these actin microfilaments. And myosin works right alongside actin to generate muscle contraction. So muscle cells have lots of myosin filaments. Myo means muscle, right? Myosin was first discovered in the muscle cells, but there's actually an enormous family of myosin proteins, which are mostly involved in cellular motility in one form or another. And muscle contraction is certainly a big part of that. Then for the intermediate filaments, let's look at number 4 in your study guide. Now, this isn't really all that clinically relevant unless you're going into pathology and you need to know which stains can be used to identify different intermediate filaments, which we're going to talk about in a second. But this is important to know for step 1. The first one in your study guide is vimentin. So vimentin is an important structural component of connective tissue, including fibroblasts, leukocytes, endothelium, that sort of thing. It's going to provide a strong support for the cellular membranes, and it keeps certain organelles fixed within the cytoplasm. So vimentin is a pretty strong type of intermediate filament. The next one's desmin. Now desmin's found in all types of muscle cells, smooth muscle cells, skeletal muscle cells, cardiac muscle cells. Cytokeratin is found in epithelial cells. Think about keratin. So this is another very strong filament. It's found in desmosomes and hemidesmosomes, which are attachments between epithelial cells and other epithelial cells, and also between the epithelial cell and the basement membrane. Dr. Lewis is going to talk about that in the dermatology videos. The next one is GFAP, or glial fibrillary acid proteins, which are found in glial cells, astrocytes, Schwann cells, and so on. It's a type of ectodermal intermediate filament. Then it's peripherin, which is found in the neurons. And then there are neurofilaments, which can be categorized by their molecular weight. So there's low, medium, and high molecular weight neurofilaments. Neurofilaments are found within the axons. There are also nuclear lamins A, B, and C. Now, these are definitely important to know for your exam. Nuclear lamins are like the skeleton of the nucleus. They provide a structural framework for the nuclear envelope. And we saw that the cyclin B and CDK1 complex was associated with the breakdown of the nuclear envelope and the nuclear lamins, which has to take place during mitosis in order for the nucleus to divide. The other thing that's worth knowing about nuclear lamins is that mutations in the nuclear lamin genes are implicated in several diseases, such as progeria. Now, progeria is a disease that causes advanced cellular aging. So the 5-year-old child might have wrinkles and the facial structure of an 80-year-old because these nuclear lamins are mutated. And then muscular dystrophy is another disorder where you could have mutations of the nuclear lamins. Now let's talk about how the immunohistochemical stains can be used to identify these different intermediate filaments. Again, different cells and different tissues are going to have unique intermediate filaments. So if you had a metastatic tumor somewhere, such as in the brain or in the liver or in the lungs, you do a biopsy and the pathologist is going to stain the tissue to try to identify which kinds of intermediate filaments were present. That's going to help you determine what kind of tissue the primary tumor came from. sometimes it's hard to identify where metastatic tumors originated from, and these stains can help you identify what the origin of the tumor is. So we said that the intermediate filament vimentin is found in connective tissue. So that stain is going to be helpful to identify sarcomas and also some carcinomas, but primarily I want you to associate vimentin with sarcoma. Then desmin is found in muscle cells. So if you have a sarcoma of the muscle cells, a myosarcoma, you could use a desmin stain to help identify that tumor. So this might be like a rhabdomyosarcoma or a leiomyosarcoma. Cytokeratin stain is used to identify carcinomas. Now, a carcinoma is a cancer that arises from epithelial cell. So again, cytokeratin is mainly for carcinomas, vimentin mainly for sarcomas. And GFAP is a helpful stain in identifying some astrocytomas, such as glioblastoma. And then again, a neurofilament stain stains neurons rather than glial cells. So it can be a useful stain to help you identify a tumor of neural origin, such as a neuroblastoma, which is a childhood tumor that arises from sympathetic ganglion cells in the adrenal medulla, and also for identifying a rare tumor called a primitive neuroectodermal tumor. All right, let's go to the whiteboard and draw this out in a table format. I want you to either take out a piece of paper and copy these down with me, or you can make notes on the table in your study guide. Now hopefully these aren't too hard, but chances are you're just going to have to sit down and memorize these. So the first type of filament we have is vimentin. We said that was seen in the tissue type of connective tissue. And the main tumor I want you to remember for this one is sarcoma. It can also be seen in certain types of carcinoma, but mainly think about sarcoma for vimentin. The next one is desmin. We said that was found in muscle tissue, so you can think about muscle tumors, such as a rhabdomyosarcoma and also a leiomyosarcoma. The next one is cytokeratin, which is seen in epithelial cells. And this was a big one for carcinoma. The next one is GFAP, which is seen in glial cells, so you can think about glial tumors like a glioblastoma. And our last one, neurofilaments, obviously seen in neurons. You can think about neural tumors like neuroblastoma and also the primitive neuroectodermal tumor. All right, I hope you did pretty well on this one. And if so, I'm going to give you 2 gold stars. The largest type of cytoskeletal filaments are the microtubules. Microtubules are tubes made up of proteins called tubulin. And there are 2 types of tubulin, alpha tubulin and beta tubulin. And first they come together to form a dimer, and then the dimers join together lengthwise to form something called a protofilament. And then those protofilaments come together side by side to form a cylindrical microtubule with a hollow core. And the way the alpha and beta subunits are arranged, they form a spiral or a helix that runs the length of the microtubule. Now, in terms of function, I want you to remember movement. Microtubules are involved with movement, a couple of different kinds of movement. For starters, microtubules are an important part of the structure of flagella and cilia, and they're what enable the cilia to move, as we're going to see shortly. Microtubules are also part of the mitotic spindles, which are what pulls the duplicated chromosomes apart during nuclear division. Now, this is a simplified explanation, but basically during mitosis, the microtubules assemble toward the chromosomes and latch on, then they disassemble very rapidly and pull those chromosomes apart so the cell can divide. Microtubules polymerize and grow very slowly, but they can collapse fairly quickly. So if something prevents the microtubules from growing and then collapsing, that'll interrupt mitosis. So, what drugs act on microtubules? Well, the first group are cancer drugs, and there are 2 different groups of cancer drugs that act on microtubules, vinca alkaloids and taxanes. So the vinca alkaloids are vincristine and vinblastine, and they work by blocking the polymerization of microtubules. So they prevent the microtubules from growing. We're going to talk about that again at the very end of the course in the oncology section. And then the taxanes or taxols include drugs like paclitaxel and docetaxel, which work by hyperstabilizing the microtubules. So the microtubules can grow, then they can't break down in order to pull those chromosomes apart. So the vincas and the taxols are similar in that they both act by interfering with microtubule function and preventing mitosis of the cancer cells, but on a cellular level they actually work in very different ways. Then, other than cancer drugs, there are benzimidazoles, which are antihelminthic drugs like mebendazole, albendazole, thiabendazole. These are all used for certain worm infections, hookworm, tapeworms, that kind of thing. You can remember that worms are bendy, so you're going to use mebendazole and albendazole to kill them. Griseofulvin is an antifungal drug that works on microtubules. It's used for ringworm and nail fungus mostly. There's also colchicine, which is an anti-inflammatory drug used to treat gout. So phagocytes use microtubules, and then colchicine helps to reduce inflammation because the phagocytes can't move to where they need to go because the microtubules aren't functioning properly. One other function of microtubules is to allow the cell to move things around within the cytoplasm. Remember, microtubules are involved with movement. They help the cilia to move. They help phagocytes move. They help chromosomes move during mitosis. Now they're helping the cell move stuff around from one part of the cell to another. For instance, some cells are really big. Take a motor neuron for your big toe. The cell body is way up in the anterior horns of the L4, L5 region of the spinal cord, but the axon stretches all the way down to the big toe. That could be 3 or 4 feet depending on how tall you are. So there needs to be a reliable mechanism for moving things from the cell body to the other end of the cell. And this process is called slow axoplasmic transport, and it basically uses the microtubules like a railroad track to guide things to the right location inside the cell. It's not just neurons. Other cells use these microtubule tracks to move things around also. So how exactly does this work? Well, the railroad cars that actually move things up and down these microtubule tracks are called molecular motor proteins. And there are 2 of these molecular motor proteins that you should know about, dynein and kinesin. Microtubules are oriented with a plus end and a minus end. The end of the microtubule made up of alpha tubulin is the minus end, and the end with beta tubulins is the plus end. So dynein and kinesin attach to the outside of the microtubule, and they also attach to whatever cargo they're hauling, and then they walk along the microtubule from one end to the other, dragging their cargo with them. So kinesin walks toward the plus end of the microtubule, which is generally going to be away from the nucleus, so that's called anterograde transport. And then dynein walks in the other direction, toward the minus end, which is called retrograde transport. We're going to come back to dynein in just a second. Let's talk next about cilia and flagella, because they're both composed of microtubules. So the flagella of sperm are structurally very similar to a cilium, just a lot longer. In motile cilia and flagella, the microtubules come together in a 9+2 arrangement. So you have a circle of 9 pairs or doublets of microtubules on the outside, and then 2 individual microtubules running down the center. And then lots of dynein motor proteins stick out from the microtubules and walk along adjacent doublets, which allows each microtubule doublet to move relative to another doublet so that the cilia moves as an entire unit. Now, where in the body do we find cilia? We find them in the respiratory tract, including the trachea and the bronchioles, and we also find them in the paranasal sinuses because cilia help sweep out mucus and bacteria and debris. The fallopian tubes are also ciliated because cilia help to sweep the ovum or a fertilized zygote toward the uterus. Now, keep that list of ciliated structures in mind for just a moment. One other thing to know about how ciliated cells work is how they coordinate their ciliary motions. So if each ciliated cell was doing its own thing, you wouldn't be able to sweep anything in any particular direction. It would just be kind of going on randomly. But instead, we see a coordinated ciliary contraction taking place that moves things in one direction. So how does one ciliated cell communicate with another ciliated cell to coordinate this contraction and create this wave-like motion? Well, it's accomplished by gap junctions between the cells. So gap junctions are important for coordinated contraction of the ciliated cells. That's a detail that's worth knowing for your test. Now, what happens if the molecular motor proteins in the cilia don't work? If the dynein doesn't work, you don't have this movement of the cilia in the respiratory tree or in the sinuses or in the fallopian tube or the flagellum of the sperm, you're going to get something called primary ciliary dyskinesia. Again, this is due to a defect in the dynein arms. If the dynein can't walk like it's supposed to, you won't have movement of the cilia. So this results in fertility problems in both males and females because in the males the sperm are immotile, and in females because the fallopian tubes aren't going to sweep the zygote to the oocyte anywhere. Now, you can also see bronchiectasis because these patients can't clear mucus out of the bronchial tree. They're going to have chronic sinusitis because they can't clear out the sinuses. And then cilia are also involved in determining the position of internal organs during embryological development. So 50% of patients with primary ciliary dyskinesia also have situs inversus, where the organs are on the opposite side of the body. So the liver would be over on the left, and the spleen would be on the right, and there's dextrocardia where the heart is on the right. Now, not all patients with situs inversus have primary ciliary dyskinesia, and not all patients with primary ciliary dyskinesia have situs inversus. But if you have the triad of sinus inversus, bronchiectasis, and chronic sinusitis, that's called Cartaginer syndrome. But again, 50% of patients have primary ciliary dyskinesia without situs inversus. So Cartaginer syndrome does not necessarily equal primary ciliary dyskinesia. When the cilia don't work, there's a 50-50 chance whether your organs are going to be on the right side or the wrong side. All right, that's it for the cytoskeleton. So what else are you going to find in the cytoplasm? Well, there's water, obviously, and a lot of things are dissolved in that water. There's electrolytes, sodium, potassium, a little magnesium, some calcium. You have enzymes like protein kinase A. That's an enzyme we're going to see a lot throughout the course. There's tRNA and amino acids that are floating around in the cytoplasm so that you can build proteins. There are sugars like glucose and fructose that are phosphorylated. There are phosphates in the cytoplasm. There are vitamins in the cytoplasm, which are often used as cofactors for various cellular processes and reactions. And there's a fair amount of cellular waste in the cytoplasm as well. And then what about some of the more insoluble cytoplasmic inclusions? All those electrolytes and sugars and amino acids and things can be dissolved in water, but sometimes your cytoplasm contains big chunks of things, some of which may be good, some of which aren't so good. Glycogen, for instance, is where we store glucose molecules. It's kind of a giant branched glucose polymer. You're going to find glycogen primarily in the cytoplasm of liver cells and also muscle cells. Then lipid droplets can also be found in the cytoplasm of certain cells, in particular adipocytes or fat cells. So most of the cytoplasm of adipocytes is just a huge lipid droplet because the purpose of adipocytes is to store lipids. So you're going to find lipid droplets in the cytoplasm of adipocytes. You're also going to find lipid droplets in the hepatocytes that are undergoing fatty change. And sometimes in the cytoplasm you can have granules of pigment, such as hemosiderin, which is an iron pigment. And then sometimes you can have pathologic inclusions in the cytoplasm. For example, a lot of different viruses will cause inclusions in the cytoplasm. Cytomegalovirus causes owl's eye inclusions in the cytoplasm. Rabies virus can cause what's called Negri bodies in the cytoplasm of the neurons. Herpes virus can cause Cowdry bodies, which are sometimes referred to as Cowdry type A inclusions. We're going to look at all those when we talk about viruses in the microbiology section. You can also have cytoplasmic inclusions in neurodegenerative diseases. For example, in Alzheimer's disease, you have neurofibrillary tangles made up of tau protein that are found in the cytoplasm. In Parkinson's disease, you get Lewy bodies in the cytoplasm. And one of the things that's been proposed as a mechanism of aging is just buildup of stuff in your cytoplasm that your cells can't get rid of. And then you'll see inclusions in lysosomal storage disorders like Gaucher disease or Niemann-Pick disease or Tay-Sachs disease. Remember, the lysosome is the place where debris and waste products get digested. And if the lysosomes don't work very well, those waste products are going to accumulate in the lysosome, but they're not going to be digested. So that lysosome is going to sit there filling up with junk. So you're going to see cytoplasmic inclusions in lysosomal storage disorders. Again, Gaucher disease, Niemann-Pick disease, Tay-Sachs disease. And also in mucopolysaccharidoses, such as Hurler syndrome and Hunter syndrome. It also happens in inclusion cell disease or I cell disease, which is where the lysosomal enzymes don't get the correct mannose-6-phosphate address label, so they can't make it to the lysosomes. The lysosomes just fill up with undigested garbage. And then there's an immunodeficiency disease called Sheidak-Higashi syndrome, where the lysosomes of the phagocytes are ineffective, and that leads to recurrent infections. But in those phagocytes, you can see lots of cytoplasmic inclusions. All right, that's it for this video. So go ahead and answer the end of session quiz questions, and then we'll go through the answers together. First question, what drugs act on microtubules? So I mentioned 4 cancer drugs, vincristine, vinblastine, paclitaxel, and docetaxel. And then we mentioned a few other drugs. There's the benzimidazole antihelminthic drugs like mebendazole, albendazole, thiabendazole, all those bendy drugs. There's griseofulvin, which is an antifungal. And there's colchicine, which is an anti-inflammatory that acts on the microtubules of the phagocytes. Next, what intermediate filaments are found in the following tissue types and cellular structures? So in connective tissue, we said that was vimentin. In muscle tissue, it's desmin. In epithelial tissue, it's cytokeratin. And in the axons, it's neurofilaments. And the last one, what are the defects seen in Cartaginer syndrome? So again, Cartaginer syndrome is a triad of bronchiectasis, chronic sinusitis, and situs inversus. And then because Cartaginer syndrome is also a form of primary ciliary dyskinesia, you can also add infertility to that list because infertility is a major feature of primary ciliary dyskinesia. All right, that's it for Foundations 4. I'll see you next time.
2 drugs with mechanisms not the same. The vincas and the taxanes are their names. One inhibits, the other a microtubule enhancer. Yet both are used to fight the dreaded cancer. As our tragic story doth begin, starring these inhibitors of tubulin, a pair of star-crossed lovers form our scene, brave paclitaxel and his vincristine.
Soft! What light through yonder window breaks? It is the east!
and then Christine is the sun. Arise, fair Vinca alkaloid, my sweet. Oh, that I were a Ewing sarcoma, and that thou wouldst inhibit polymerization of my microtubules and disrupt my mitotic spindles.
Oh, paclitaxel, paxlitaxel, wherefore art thou paclitaxel? Oh, what's in a name? That which we call a taxane by any other name would smell as sweet.
Why must our families oppose one another this way?
It is our very nature, my love. We Vincas inhibit cell division by inhibiting microtubules, while you taxanes stabilize microtubules.
Yes, but the end result is the same. Oh, why must this keep us apart?
Besides, you taxanes cause me such devastating hypersensitivity reactions.
Premedicate! Premedicate, my queen! With a steroid and an antihistamine!
Ronnie, this isn't working.
No, you're doing great! This show must go on!
This is a terrible way to study. I don't have time for these stupid role-playing games of yours. Can't believe I put on a corset for this.